All right. So let's talk about the adaptive immune system, right? We had a brief kind of overview of the innate immune system. I didn't mention it, but sometimes in the past you would have seen the innate immune system referred to as the non-specific immune system, the adaptive referred to as the specific. I tend to not like that terminology because I think it does not give the innate immune system a lot of credit. And hopefully, what you can appreciate from yesterday is toll-like receptors, nod-like receptors, rig-like receptors, right? Those are looking for patterns, but they're looking for specific patterns. And so their response is targeted towards pathogens, is very efficient and effective, right? And so non-specific is I think a dying term, right, as opposed to innate. So adaptive immunity, and we've actually mentioned this in some of our articles. I can't remember who presented it, but it was one of the early ones where they were looking at the differences in soil, the microbes present in soil between, I don't know if it was Russia and Finland, whose article that was, and trying to associate that with a difference in the prevalence of autoimmune diseases and allergies, right? And so, if you look down here, the hygiene hypothesis came out in 1989, right? And this was based off of observations of a difference in the number of autoimmune cases and allergy cases in East Berlin compared to West Berlin, right? East Berlin, which was communist and not as good at living conditions, they had less issues with allergies and autoimmune diseases compared to West Berlin, which was the democratic and free area where life was a little better. All right. But, right, if I jump back up, sorry, going a little out of order. One of the things about adaptive immunity is, right, it's highly specific, right? If we go like to toll-like receptor 4, it's responding to LPS, right? So all gram-negative bacteria have LPS. And so in that case, it's not distinguishing different species of gram-negative bacteria. It's just saying this is a gram-negative bacteria, we have to respond, right? We start to see an adaptive immunity that the response towards a specific pathogen is highly concentrated and focused on that individual pathogen. Right? So here we're saying include cells of mononuclear phagocyte system and cytokines. So this is a fancy way of saying macrophages and dendritic cells and the signals that they secrete are important in activating and communicating with the adaptive immune system. Another important thing, and we'll touch on this, is that the adaptive immune system has to be taught how to tolerate and protect our own cells while identifying pathogens, right? When we talk about the innate immune system, all right, the information that it can understand and communicate with the rest of your body is inherent, right? That's part of when you're born, right? You have your innate immune cells that have their pattern recognition receptors and they're ready to go, right? The adaptive immune system is a more of a education lifelong process. And so what we see here are some of the cell types that are important in adaptive immunity. And note, right, at the top, here you have a dendritic cell. Remember, you could also add a macrophage to here, although dendritic cell is better at communicating with the adaptive immune system. Macrophages can also do this, all right? And so you see this dendritic cell is communicating with T cells. We'll talk about these different types of T cells here in just a second. The communication from the dendritic cell to the T cell allows them to become active and do their job. Again, we'll go over that in more detail here in just a second. You also see here B cells, and although the B cells are not directly communicating with dendritic cells, right, we will talk about B cells' ability to communicate with T cells, which is needed for their activation. And if you've heard of antibodies, which I'm sure you have, but maybe not a lot of detail, but I think you guys should have probably read about those or looked over some of those, maybe with your assignment. Antibodies are being produced by B cells, and we'll talk about different ways that they neutralize pathogens, which I know was one of your questions on your mastering assignment. All right. So this chart is incredibly simple, but it delivers, I think, a powerful message. And the message that I love from this chart is it is telling you and showing you that although, right, you have two branches of the immune system, and one is maybe more specific, right? If we're talking about the adaptive than the other. They both are essential. And so here you see this red line, right? This is showing, and notice here, duration of infection on the x-axis, number of pathogens present in the system on the y-axis. And here, if you lack an innate immune system, remember your innate immune system is your first line of defense, right? So you don't have that first line of defense. If you lack an innate immune system, you're also lacking those macrophages and dendritic cells that communicate with the adaptive immune system. And here the pathogen would go unchecked, all right? And notice, right, you could have this increase in pathogen load, you could still have an adaptive immune system, but without the innate immune system, it can't do anything for you, all right? Our next line is the green line, and notice here that this is saying you're lacking adaptive immune system only. And so you see that you don't have this exponential increase in pathogen load. And the reason you don't have that increase is because your first line of defense, right, that innate immune system is doing its job. But to finish off and thoroughly fight an infection, you need adaptive help. And so although you're able to keep the pathogen load down, eventually over time, right, and we could extend this graph and you would see it start to kind of trickle all the way up, all right? What's really interesting, we'll talk about this a little bit more, right? This is almost kind of what happens in HIV. It's not that they lack an adaptive immune system, but the virus starts to kill their adaptive immune system, right? And so they become very susceptible to common infections that their body deals with initially, but then cannot ultimately control, right? And here's where I hope most of us fall in, right, is into the, I like this, normal humans. I guess that's a very relative term, right? I don't know if I would use normal for all of you, but for most of you, all right? And so here, right, the innate immune system responds, helps keep the pathogen load down, the adaptive immune system is activated, and it finishes it off, okay? So again, you need both of these systems, and this chart is a good representation, although simplistic representation, of why that's so important. Okay. So let's talk about, with the adaptive immune system, you're going to have two branches, right? So if anything, with any type of biology or ology, you notice there's a lot of branches, and I'm sorry, they just don't stop, right? So your immune system has two branches, innate, adaptive. The adaptive immune system then has two branches, cellular and humoral, all right? So when we talk about cellular immunity, that is referring specifically to T cells, okay? And again, we'll keep on revisiting T cells and the role that they play. But for T cells, there are two major types of T cells, see more branches, that we are really interested in. And I'll point out some other T cells, and even within these two types of T cells, there are a lot of variations. But for simplicity right now, we'll say that you have cytotoxic T cells and you have helper T cells, all right? And again, I'll talk about how those T cells are different and the different roles that they play in just a little bit. T cells, no matter what type you are, have something called a T cell receptor. And you actually see that on this diagram here. Notice next to that T cell receptor, you have something called a CD marker. So maybe if you've heard of CD4 or CD8 T cells, right? This is a protein that essentially helps stabilize the T cell receptor, and the CD marker is specific for different types of T cells, right? And so importantly, right, this T cell receptor is going to bind very specific antigens or portions of an antigen. And so one thing I want to point out is, like, let's say you have influenza, right? So you have the flu and you also have strep throat, all right? The T cell receptor that is going to recognize pieces of the influenza virus is going to be very different from the T cell receptor that's going to recognize parts of the strep bacteria that's causing strep throat. All right. And so these are highly specific, right? They're not like the pattern recognition receptors. Although they look for patterns, they look for very, very specific patterns. It's important to note that T cells will not bind free-floating antigen. And the reason I want to make you guys aware of that, and you see that here, is that the receptor, right, of another cell has to present the antigen to the T cell receptor. All right. So that would be, you know, like let's say you're doing an Easter egg hunt. All right. And Megan's the person I see on my screen, so I'll use Megan, right? And so there's all these eggs out in front of Megan, and she can't do anything about it. She can't pick them up unless someone who's with her picks that egg up and hands it to her, and then she can interact with it, right? So T cells require the presentation of the antigen in order to interact with it, all right? And you'll see in just a second, this is very different from B cells, all right? So for T cell activation, right, this is a two-step process depending on where you're looking in the research literature. If you were ever to dabble in that, you would see that there are multiple groups that would argue a third signal for activation, and that's fine. I'm not saying that's not true, but the two most important steps for activation are recognition of the antigen, all right, and then upon recognition of that antigen by the T cell receptor, the cell that is presenting that antigen will release stimulatory signals to cause the activation of that T cell. All right, so you need to recognize the antigen that's being presented, and then stimulatory signals need to be released to activate that T cell. And again, we're going to go over this in even more detail, but I kind of want these seeds to be planted. Okay. So for the different type of T cells, and again, we'll continue to revisit this, but cytotoxic T cells, and I'll mention this later, also called CD8 T cells, if maybe you've heard of that before. These T cells, right, are going after infected self cells, all right? So a lot of times, the majority of the cells that cytotoxic T cells interact with are cells that have a virus in them, right? Because viruses are very dependent, right? We talked about this on hijacking the host cell machinery. And so cytotoxic T cells are looking for these infected cells and then are killing them via apoptosis, right? So the idea would be if you're a cell and viruses are being made in you, right? You become a virus factory. The best way to stop the virus then in some cases is to destroy the virus factory, which happens to be your own cell. Okay? We don't really go into it much here, but cytotoxic T cells are also just great at recognizing cells that look abnormal or are behaving abnormally. And so if you've heard of anybody that has had T cell mediated treatments for cancer, that is going to be cytotoxic T cells that are being used there. And even in our bodies, right, cytotoxic T cells are important in patrolling and hopefully preventing cancers from starting. All right. You'll see this theme, and I want you to hold on to this because this is going to be important now and even more important when we talk about vaccines, is that some cells become memory cells. What you'll see in the innate immune system, or not in the innate immune system, I'm sorry, in the adaptive immune system, is the idea that you have effector cells. Those can be T cells, those can be B cells. Those are the cells that are actively fighting the infection. Essential. You want them there because they remove the infection. What we also see, though, is that a group of cells are essentially set aside that have memory of that same pathogen. And so when that pathogen comes back, these memory cells start the immune response much more quickly than it was started before, hopefully preventing the infection from taking place. And that is the fundamental idea of vaccines that we'll revisit in a little bit. For T helper cells are producing cytokines that activate B cells and macrophages, and you can even put on here can lead to the activation of cytotoxic T cells. Okay, so one thing to appreciate early on here is, dendritic cells and macrophages are a key component in linking the innate and the adaptive immune system. And with those two being linked, okay, you could argue that the T helper cell is kind of the keystone or the linchpin that helps hold everything together. The signals that they release lead to activation of both the innate and the innate and the adaptive immune system, right? Like cytotoxic T cells, during an active infection, some of them will focus on activating cells that are fighting the infection, while some of them will be set aside to become memory cells. Again, we'll revisit this. This is what makes HIV so deadly. HIV likes to target T helper cells. And when you remove T helper cells from the system, right, you are removing your ability to activate B cells, you're removing your ability to activate macrophages and dendritic cells, and then the whole immune system falls apart. All right. Another cell type that we won't really talk about too much here is something called a Treg, right? A T regulatory cell. As tempting as it may be, don't call them Tregs. I had a friend in grad school who did Treg research, and we were at a presentation. I was at a presentation that he was giving, and one of the panelists asked him something about Tregs, and it took us both a minute to realize that she was talking about T regulatory cells. So I'm used to it now, so I guess you want to call them Tregs, that's fine, but they're called Tregs, right? And they suppress immune activation. So if you had Tregs that were dysfunctional, right, or maybe not present in a high enough quantity, Amanda, what type of problems might you have, do you think? And they were actually mentioned today.
Would it fail to turn on your immune system?
Well, if it suppresses it, right, it's going to fail to turn it off. So what type of disorders or problems arise when you can't turn off your immune system?
Autoimmune diseases?
Autoimmune diseases, right? Amongst many things. And so what we've actually seen, and type 1 diabetes is my favorite autoimmune disease. But what we know in type 1 diabetes is that the number of T regulatory cells that is present in those patients is much lower than in someone that has a normal immune system. And so, without Tregs, your immune system might get out of control or out of hand in response to an infection and then potentially lead to an autoimmune disease. But that doesn't happen because Tregs are there and they help kind of dampen that response, right? Or make it end, right? They put an end point or stop point on it. Just like you have the M2 macrophages that are good at suppressing, Tregs kind of help rein in an immune response. And we know in a lot of autoimmune diseases, there's either dysfunctional T regulatory cells or the abundance of T regulatory cells is not as high as it should be. All right. So that's cellular immunity, right? That focuses on T cells. And again, note that cellular immunity and humoral immunity, right, are going to be tightly linked. Humoral immunity, if you've read, any, I don't know why you would, but if you heard of, the ancient Greeks and their medicine, right, this idea of humors, which are basically different body fluids, right? And B cells, right, are a lot of times going to secrete antibodies into body fluids. And so that's one of the reasons that we call it humoral immunity. And so B cells, right, again, are activated and divide upon antigen recognition. And so again, you see this common theme in both T cells and B cells is that there needs to be antigen recognition. And then upon antigen recognition, there needs to be some type of activation. For B cells, all right, the effector version of a B cell, the B cell that's actively helping to fight an infection. This is called a plasma cell, right? And you see down here, the plasma cells are producing the antibodies. There are effector T helper cells and effector cytotoxic T cells. We don't have cool names for them, right? We just call them effector T helpers or effector cytotoxic T cells. But for B cells, the effector cell is called a plasma cell. Like T cells, right? You have some that are fighting the infection, the plasma cells, but then you have some cells that are set aside and become memory cells, right? So that they can wait around and respond to that same pathogen if it were to come back. All right, so as just mentioned, plasma cells activate B cells that produce antibodies. B cells have a B cell receptor, just like T cells have a T cell receptor. Now, structurally, I'll point out, and we'll see that they're a little different. And in the case of a B cell receptor, if you've ever seen an antibody before, and again, I'll show you what one looks like in a second, a B cell receptor is essentially an antibody that is bound to the cell membrane, right? When we talk about antibodies, we talk about things that are floating around in the system, but a B cell receptor is an antibody that's bound to the membrane. And note, these B cell receptors can bind free floating antigen. So they don't have to have something that's presenting that antigen to them, right? T cell receptors need that help. They need the antigen presented to them, while B cell receptors can bind to that free floating antigen, right? And importantly, and we kind of mentioned it up here, this idea of activation. So it's not just enough to recognize an antigen, whether you're a B cell or a T cell, you need help. And B cells are specifically getting their help from T helper cells, again, emphasizing or highlighting the importance of the T helper cell. Okay. So as long as we're talking about humoral, immunity, let's talk about antibody structure. So the first thing you probably notice, and maybe you've seen this before, is that an antibody is kind of shaped like a Y. And here you can see these are 3 different images that are just showing kind of the different ways that we divide this structure up. Here you have an FC region, and here you have a Fab region, all right? The FC region of antibodies are identical, no matter what type of antibody you are. And we'll talk about the different classes in just a second. Where antibodies get their variability or their variety is in this Fab region. And so here, right, you can see antibodies divided into heavy chains and light chains. And you can see heavy chains red, light chain are these fun colors, right? And you can just say, well, heavy chain, light chain, there's just more heavy chain, right? So that's actually more weight, right? More protein weight. And here within the light chain, the ends of the light chain, you have these antigen binding sites or these variable regions that allow antibodies to bind to specific pathogens, right? Remember, very specific. So an antibody that binds to the influenza virus is going to be, very different than an antibody that binds to the HIV virus or, right, the COVID-19 virus, right, SARS-CoV-2. So very, very specific constant regions. One of the things we'll point out here with these FC or these constant regions is that cells of the innate immune system have receptors on their surface that bind these constant regions. Okay, so this is just highlighting kind of my point that antibodies in the adaptive immune system are very specific to pathogens. And not only do they respond differently to something like the flu, HIV, SARS-CoV-2, right? You have different antibodies for all those different pathogens. Even the same pathogen might have different antibodies that are binding to it. And what do we mean by that? Well, if we talk about antibodies binding to antigens, right? And an antigen is a part of a pathogen. Specifically, within antigens, you have a region called an epitope, right? And an epitope is what allows the antibody to bind to the antigen. And this is a bacteria cell, right? You can make it up or call it whatever you want. You can say this is S. aureus, right? We'll say it's S. aureus. And you see S. aureus, right? These little green things here are the epitopes of different antigens. And so they show you different shapes. So here's a square, a cone, and a sphere, all right? And here you have an antibody that is recognizing the cone. Here you have an antibody that is recognizing a sphere, and presumably one of these other antibodies that's floating around here maybe recognizes the square, all right? Although they are recognizing the same pathogen, they are recognizing different regions of that pathogen, all right? So the antibody that binds to the cones on S. aureus is not going to bind to S. aureus via the spherical antigens, okay? And so one of the things that I'll probably point out in the next lecture when we talk about some of the COVID-19 vaccines that are being developed is they're all going after the same virus, right? But they're going after that virus in different ways, all right? And this is really beneficial to the immune system. Why is this beneficial to the immune system to have multiple antibodies that target the same pathogen? What's something that we actually talked about in the genetics unit with bacteria that would maybe tell you that it's important to have multiple antibodies for the same pathogen? Does anyone remember?
So they wouldn't have like resistance, because like, you know, bacteria with like antibiotics, they can become resistant. So having multiple would be hard to like be resistant to all of the different antibodies.
Okay. So I like the concept, right, that you approach there. You know, antibiotic resistance is a little different, but the way you're approaching is the same general principle. I like that. So if you remember, we talked about antigenic variation. So I used that picture of Bill Murray when he was dressed up like a Ghostbuster, Bill Murray when he was dressed up for Life Aquatica, and then Bill Murray in the suit, right? And so it's Bill Murray, Bill Murray, Bill Murray. All right. But he looks different. And every time he looks different, the immune system has to generate a new response. And so in this case, if you have multiple antibodies that are binding to or looking for different antigens of the same pathogen, it makes it easier for you to remove them, even if they're trying to change things. Because maybe the bacteria tries to change the antigen that's represented here by the cone. Well, that's fine, because I have an antibody that's recognizing the sphere antigen, or I have an antibody that's recognizing the cube antigen, all right? And then vice versa. When it's trying to change these, you have other antibodies that are recognizing different parts of that pathogen. And so it is kind of ensuring that you have alternatives to recognize that pathogen in case it does something kind of crazy or tricky to change on you, okay? And so antibiotic resistance, to Noel's point, the mechanisms are different, but the same concept is true. And so one of the things that, who's doing tuberculosis for their disease? I know someone signed up for that.
Rachel D.
Okay, so Allie and Rachel D, right? And so one of the things, and this is not going to be a spoiler for their presentation, but one of the things I'm hoping they'll talk about, and if they weren't, they will now, right, is this idea that for tuberculosis and any type of mycobacterium or mycoplasma, often treatment of them entails the use of more than one antibiotic, all right? And the reason that you're using more than one antibiotic for those pathogens is because you have to use antibiotics for such a long time. And if you use more than one, it limits their ability to develop resistance, all right? And so Noel's right in the general principle, although the mechanisms are a little different, right? So you see that repeated though. But, right, more than one is good, because to Noel's point and to the point right here, it's going to help limit the ability of that pathogen to develop resistance, right? So what are antibodies doing, all right? And you guys had a question that I believe touched on this, or a section that touched on this on the assignment that you hopefully completed. And so we know, right, a lot of you probably heard, hey, when I go and get a vaccine, you know, I get protection, and antibodies are produced when I get a vaccine, and those antibodies are important. And I totally agree. But let's talk about what they're specifically doing, all right? And, you could group these 6 things into 2 major categories, right? You are either tagging them for removal from the system, right? And or preventing them from interacting with their target cell. Okay, so let's talk about these in a little more detail. So here you see this idea of neutralization. All right, viruses, remember, are really, really small. And so in this case, what it's showing is that you can have multiple antibodies that bind to a virus and neutralize it. And when you neutralize this virus, when you have antibodies bound all over it, you prevent those spikes that are present that we mentioned before from finding their target receptor on whatever tissue they want. All right. One of the things you see here is that some bacteria actually produce toxins. All right. And so if you neutralize the toxin, that prevents tissue damage from occurring or from some type of illness from occurring, and gives the immune system time to remove the pathogen that is causing that toxin to be produced. So those of you that have hopefully had your tetanus shot, which probably came in the trifecta of your DTaP, the tetanus bacteria itself, hey girl, the tetanus bacteria itself doesn't actually do that much to you. It's the toxin that it produces that has a negative effect on neurotransmission and then muscle control. So if we neutralize that toxin, then we prevent the pathogenicity. So some of you have stepped on a rusty nail or gotten a bad wound, and you hadn't recently had your tetanus shot. One of the things they'll do for you is they'll give you your tetanus vaccine, and then they'll also give you a dose of antibodies that recognize that toxin. And we'll talk about that more in the immunity vaccination unit. All right, opsonization. You guys have already kind of seen this a little bit, but the idea of, again, a bacteria is coated with antibodies on this phagocyte, which could be a dendritic cell or macrophage. You have FC receptors. They bind to the FC region of the antibody. We engulf this bad boy, and then you guys have seen what happens. It eventually results in a bacteria being broken up and essentially pooped out, right, with some of the antigen presented on the surface, which we'll go over more. We talked briefly about complement activation, and so the idea that the classic pathway is antibody dependent, and so antibodies actually interact with the complement interacts with the FC region of the antibodies, leading to the activation of that complement protein, right, which eventually results in C3 convertase being made, and then we know what happens from there. Immobilization and prevention of adherence. And so again, you can bind to antibodies to the fimbriae or antibodies to flagella. If you bind them to fimbriae or flagella, right, you can prevent them from either traveling to where they want to go, or you can prevent them from being able to attach to the receptors they want to bind to. Cross-linking is basically a precursor for some bacteria before opsonization. And so this idea being, if I were to take, if any of you, when you were younger, used to do those like dive sticks in the pool. Sarah, did you ever do the dive sticks in the pool? No? Laura, did you ever do the dive sticks in the pool?
Yeah, I did.
Good. Come on, Sarah. I know what I'm getting. Sarah, when are you graduating?
After this class.
After this class, okay. Well, so I'll have to stay in touch with you so I can send you some dive sticks as a graduation present, right? But, right, if you ever played dive sticks when you were a kid with your friends, you know, one of the challenges you would do is you would take those dive sticks and you'd throw them all over different parts of the pool, right? And so I know, like, me and my brother, hey, Emmeline, I know my brother and I would throw them all over, and then we'd see how many we could get in one breath, right? And it was pretty challenging if you had a big pool. You had some in the shallow end, you had some in the deep end, to try to get them in one breath, right? That's a lot of work. What would be easier and a way to cheat is to all just throw them in the exact same place. Then you go down once and you go down once and you pick them up, right? Quarantine with a 19-month-old, right? You want to know fear. You come hang out in my house for a little bit, right? But you put them all there at once, you dive down, you get them all. A lot easier, a lot less work. And that's the basic idea of cross-linking, is these antibodies basically help these bacteria or other pathogens cluster together. You have a phagocyte come in, and now all of a sudden in this picture, right, is able to eat 4 at once, and take care of them instead of neutralizing one at a time, right? So this is much more efficient, all right? Allows you to remove a lot of pathogens at once. The last one that we don't spend a ton of time on here, but we will revisit this, I think, one more time in this lecture, is this idea of antibody-dependent cellular cytotoxicity. I'm fine if you call it ADCC, but please, at least get this acronym right. So for the antibody-dependent cellular cytotoxicity, basically what happens, and we'll talk about why this happens a little bit later, but if a cell is not doing right, here you see it's infected by a virus. Sometimes even cancerous cells will have antibodies that attach to the outside of them because they are a little different, and the body starts to figure this out. And natural killer cells, which we don't spend a lot of time on, but are part of the adaptive immune system, will basically come around and say, if you're a host cell, right, and you're covered with antibodies, something's wrong, all right? This isn't normal. And so they deliver, and I love kind of these terms, something called a kill package, which is basically a series of enzymes and proteins that cause cell death, all right? So they're kind of out there patrolling and looking for abnormal cells. It's not normal if your cell's healthy to be covered in antibodies. And so these natural killer cells will neutralize those cells and remove them from the system, okay? Let's see how much battery I have left. Oh yeah, running strong. So for the different antibody classes, there are 5 different antibodies. You can see here that I only have 4 listed. It's not that I don't care about IgD. It's just that we don't really have a great understanding of what it does. The only thing that we know is if a B cell has IgD on its surface, we know that it's a mature B cell. But as far as it having a direct role in fighting any type of pathogen, as far as I know, and again, maybe something has come out recently, there's not a known role for it in the immune response, right? So that's why we focus on IgM, IgG, IgA, and IgE. Now, remember, right, these antibodies have kind of different behaviors as far as where they are at in the body, and even in some cases the pathogens that they're interacting with. But right, that FC region is going to be relatively conserved amongst all of them, right? So IgM is the first antibody that's produced when a pathogen shows up, right? And we'll look at some kind of charts here in just a little bit that show the body's response to a newly acquired pathogen. And so IgM is the first antibody that's produced to help respond to that pathogen. IgM is really cool because it forms a pentamer. So basically you have 5 antibodies that are joined together by the FC region. And so if you can kind of imagine it, and I'll show you a picture of what it looks like here in just a little bit. It looks like a little ninja star that's floating around in your blood. And so you basically have this structure that has 10 different sites on it where pathogens can bind. It's really important in controlling blood pathogens. Interestingly enough, there's a genetic disorder where patients don't produce IgA, and they have these patients that lack IgA actually have a relatively normal immune system because IgM takes the place of IgA. So kind of a cool adaptation by certain people's bodies. All right, IgG, if you guys are talking about antibodies with each other or to your friends, right, or to your dogs or your cats, whoever you like to talk about antibodies to, this is probably the antibody that you're referring to, all right? So you can see here 85%. So the majority of circulating antibodies in your body are IgG. IgG antibodies have the longest half-life, right? Like everything else, antibodies are proteins. There's protein turnover, right? That's just a normal part of life. IgG has the slowest turnover of antibodies, so they're present for a long time, right? IgG can be present for months. This is the only antibody that is passed on from the mother to the fetus, all right? So, one of the important things that's protecting that baby when it's inside of mommy, right, is the IgG that's crossing that placental barrier, right? That's going to protect the baby from hopefully any type of pathogen that the mom may encounter that may make its way to the baby. Okay. IgA is actually the most abundant antibody in your body. Most abundant antibody in your body. Yes, tongue twister, right? It rarely enters serum. It actually is mainly a mucosal antibody. So in your respiratory tract, in your digestive tract, in your urogenital tract, this is the antibody that is present. So if there's a mucosal surface, this antibody is in that surface. To give you an idea, well, antibodies are super, super small. And every time you go to the bathroom, right, you are, every time you go poop, right, you are pooping out grams of these antibodies, all right? They are secreted into the mucosal surfaces. They bind to or neutralize whatever pathogen they're associated with, and then they are expelled from the body, all right? This is also, right, the antibody that is passed on from mom to baby in breast milk, right? So remember when we were talking about, I think maybe it was Glory's article about the vaginal, swabs. We're talking about different birth options and all that fun stuff, right? So when a mom is breastfeeding, right, I mentioned this, the IgA that infant ingests helps shape its gut microbiome to hopefully something that's healthy and is good for the immune system. IgE, those of you that have allergies, this might be your least favorite antibody. IgE actually binds to mast cells and basophils, and it looks for, unfortunately, those of us that have allergies, it binds to allergens, and when IgE binds to allergens, and if it's bound to mast cells, it causes something called cross-linking to happen, and when that happens, mast cells and basophils release histamine, which causes all the unpleasantness that's associated with allergies, right? And when I say bound to, that means that the Fc region of that antibody is bound to the mast cell or the basophil, and so you have your variable region, right, that kind of Y shape that's facing out, an allergen bind, causes cross-linking, and those cells do their things. These are also, though, the antibodies that are associated with parasitic infections, all right? So they're helping to fight parasites, like if you'd have some type of protozoa in your system or some type of worm, okay? So those are the different classes. This is just a chart that I happen to love because it shows the importance of that mother-baby relationship. And so what you see here is the amount of IgG present, all right? And here you can see the dark blue is just the total IgG that's present in both the mom and the baby. And essentially what's going on here is it's telling you, right here you can see, months, 9 months, baby are born, right? If that's news to you, then please take A&P2 before you graduate, right? We'll talk about that there. But here, essentially what you're seeing is that the IgG that's present in the mom is also present in the baby, right? So it's kind of a shared system here because that mom is passing on IgG. After birth, though, right, mom is no longer producing IgG for the baby because the baby's not connected by the placenta anymore. However, you see here that we can still measure maternal IgG. And what do you know? As maternal IgG continues to dip, right, half-life, they begin to kind of go away. The baby's immune system, though, starts to take over and produces its own IgG. And so maternal IgG, you can see here, it's around for months. So that maternal IgG stays around long enough for the baby's immune system to start to kick into gear so that they can produce their own IgG. All right, so it protects the baby while the baby's inside mom before it can produce its own, and then maternal IgG remains even after birth for a long enough period of time that the baby can start to produce their own IgG and take over as the mother's IgG dissipates from their system. It's kind of a cool link between mom and baby. All right. So here we're at 9:05. So let's come back at 9:15 and we'll continue to work our way through the adaptive immune system. All right. Cool. So we've been, talking about T cells and B cells, and so now we'll just kind of focus on them a little bit more again and revisit this chart that we had initially shown for the innate immune system. So if you remember, you have your hematopoietic stem cell right here, right? These are present, and I know it was listed, but I didn't mention last time, right? These are present in bone marrow. So you have your common myeloid progenitor, which leads to the cells of the innate immune system that we talked about yesterday. And now what we're focusing on today, right, is this common lymphoid progenitor that's going to lead to the formation of natural killer cells, which again, we won't talk about too much, but then focus on T cells and B cells. And please, again, notice at the bottom, right, that they're showing that these macrophages and dendritic cells are an important link, right, for the relationship they have with the adaptive immune cells. Okay. So, again, dendritic cells, you could also add macrophages right here, are part of the innate immune system, but they are essential in T cell activation. And these cells are often the first on the scene, right? Macrophages, dendritic cells are going to be at a infection site or a site of infection before T cells are, right? So they are essential to identifying the pathogen and identifying the infection site. We talked about B cells, right? They're going to make antibodies and form memory cells to help fight the pathogens and future encounters with that pathogens that dendritic cells and macrophages find. You also have your helper T cells, which again, we said stimulate macrophages, stimulate cytotoxic T cells, activate B cells, right? So kind of that linchpin of both the innate and the adaptive immune system. And then cytotoxic T cells, right? We talked about this, inducing apoptosis in virally infected cells. And if we were talking about cancer, these cells are essential in destroying cancer cells also. All right. So of these different cell types, specifically the adaptive cells, right? The specificity that they gain, right, is through education. Remember, we mentioned adaptive immune cells have to be taught how to tolerate self and identify self versus pathogen, right? You want to be able to respond to what needs to be responded to and not respond to yourself. If you respond to yourself, right, it's a hallmark of autoimmune diseases, right? So we don't spend a lot of time on this. Those of you that are going to be around this actually upcoming fall, I believe that Dr. Christman is teaching immunology, and you will go through this in more detail in immunology if you are interested, right? But regardless, right, this is an important concept for us to at least be aware of, right? So when it comes to T cells and B cells, you have kind of two possibilities. You have negative selection for B cells, and then you have positive and negative selection for T cells. So in the case of negative selection, right, after a B cell has its B cell receptor on its surface, it's tested with self-antigen, right? So we see that the body likes to test these cells. And if that B cell recognizes self-antigen, right, the B cell receptor, if it recognizes self-antigen, apoptosis is induced in that cell, right? It's basically saying that B cell is recognizing the host, and the B cell should not be recognizing the host. So, right, we need to remove it from the system. For T cells, we have positive and negative selection because T cells have to be able to recognize MHC. So MHC is that receptor on the surface of the antigen-presenting cell, okay? And so if the T cell receptor can't recognize MHC, it doesn't matter what's in MHC because the T cell won't be able to see it. So a T cell receptor has to be able to recognize, right, that receptor that's being presented by the other cell. So that's the positive selection. So you want recognition there. Negative selection, right, then follows. And so now that we know it can recognize MHC, we put a self antigen or a self protein, right, into that MHC molecule, and we present it to the T cell. And if the T cell recognizes a self antigen, right, or a self protein, well, then apoptosis is induced in that T cell also. All right. Note, autoimmune diseases are a thing. This is not a perfect system, right? Some cells, right, in some ways you could say fall through the cracks. Another thing that becomes increasingly complicated is that some antigens on pathogens, the amino acid sequence is very similar to the amino acid sequence that some proteins have that we find in our bodies. And so the immune system can sometimes get confused, right? It thinks that it's binding a pathogen when it's actually binding self, all right? So it definitely is not a perfect system. Clearly, cells respond to self-antigen, and so we have autoimmune diseases. But for the most part, if you have a normally functioning immune system, your body is doing a pretty good job at catching any cells that are recognizing self and removing them from the system, all right? So assuming that we recognize a foreign antigen, how do we generate a response capable of fighting a pathogen and removing it from the host, all right? And that's where this idea of clonal selection and expansion comes in. And this occurs in both T cells and B cells. The chart that we're going to focus on here in just a second is looking at B cells, right? But note that a similar process is also occurring in T cells, and I will try to point that out. One of the things, and I will just give you guys a heads up, that occurs is it's possible that I could have a question on your next exam on clonal selection and expansion of lymphocytes, or clonal selection and expansion of cells in the adaptive immune system. I don't know exactly how I would word it. And it would be a mistake to just talk about how this happens for B cells in your exam, or a mistake to talk about how it just happens for T cells, right? You can still point out the general process and then mention, hey, this happens in both T cells and B cells, but I guarantee if you talk about just one cell type, you are not going to receive full credit, right? And so it's important to note that this is happening in both T cells and B cells, even though the chart that I show here focuses on B cells, all right? So let's go through these kind of 4 basic steps, or you could even call them 4 basic checkpoints. And again, this is true for T cells and B cells. So first, they start off as immature lymphocytes, all right? Kind of the most general basic definition of an immature lymphocyte here is a T or B cell that is not yet expressing its B cell receptor or T cell receptor on the cell surface, all right? So they aren't expressing T cell receptors or B cell receptors yet on the cell surface. Once they express those receptors on their surface, whether a B cell receptor or a T cell receptor, we then consider them naive lymphocytes, all right? So the receptors are there, they're ready to go. They just haven't seen an antigen yet that they recognize, okay? Upon antigen recognition, as long as it's the correct antigen, or if we're even talking about, let's say they've gone through the education process and they're hanging out in a lymph node, right? In that case, right, they bind an antigen and they become activated. Now, one of the things that I will point out here is that, remember, the adaptive immune system is not just doing things on its own, right? It needs help. And so upon antigen recognition, you also need to receive an activation signal from another cell. And we'll talk about who's activating who in just a second to remind you of that. Once these cells have been activated, antigen recognition, they receive the activation signals, they then begin to divide. And as they divide, we produce effector lymphocytes. Again, we have effector T cells that are cytotoxic, or helper T cells, We also have the effector B cell, also called a plasma cell, right, that are directly involved in fighting that pathogen. During the division of these activated lymphocytes, not only do you get your effector cells, but you also get your memory cells, right, that are going to hang around and be present for future encounters with that pathogen to generate a rapid response. All right. So this is what this looks like. So here you can see kind of your different, so here's your immature, your naive, your activated, and then you have your plasma and memory cells that are produced. All right. So here's your hematopoietic stem cell. You start here at the top. All right. Here is your immature, in this case, B cell is what it's showing, but the same general principle would apply for a T cell. It's not expressing its receptor on the cell surface, right? However, it eventually does express a receptor. In this case, we're looking at B cells, so it's a B cell receptor. But if we're talking about T cells, it would be the T cell receptor. So we now have a naive cell, right? That receptor is present on the surface. Please note here what they're trying to emphasize is that you have a B cell W, X, Y, and Z, and then you have antigens that are associated with those different receptors. Here you have antigen X. Well, antigen X is only going to interact with B cell X. All right, and so that means this is important because you're only going to be multiplying, right, the lymphocytes, whether it be a B cell or a T cell, that is needed to fight that infection. All right, if B cell X fights antigen X, well, you don't want a lot of copies of B cell W because that's not going to help you with antigen X. And the same basic principle for T cells, right? T cells will recognize, right, or interact with specific antigens and then fight the pathogen that would produce those antigens, all right? So you have your naive cell, it has receptors on its surface, an antigen binds to that receptor, in this case for B cell X. Again, this could be the same, it's the same for the same general principle for T cells. All right, what it doesn't show here, and it does write about here and here, is that it's cool, right? We recognize our antigen, that's fine, but then we also need to get help. And so in this case, right, the B cell received help signals from a T cell, right? And so the activation or help signals from the T cell induce proliferation in the B cell that has recognized antigen. And now you get clonal expansion. All right. And during this clonal expansion, you start to divide. You make plasma cells that are producing antibodies. All right. And then you also are producing memory cells that are going to be present in the system in case that pathogen's encountered again. You know, just a quick little side note, I pointed out or mentioned IgM being a pentamer and kind of looking like a little ninja star. And so here you actually see at the bottom, you can count 1, 2, 3, 4, 5 molecules of IgM, right? They're bound by the Fc region, and then you have all of these variable regions around the outside of the antibody that can potentially bind to pathogens. All right, I want to go back up quick here because, you know, the T cell follows a similar process, but note for the activation of a T helper cell, right, that's going through this same process, what's going to activate that we've already mentioned and shown us on the diagram? Elaine, do you remember what's going to activate a T helper cell?
Isn't it something released from the I don't remember.
Okay. Can anyone help her out? That's okay. We haven't really talked about it directly. Yeah.
Does the T helper cell like kind of activate itself?
Nope. Self-activation is a thing. We haven't really covered it too much. It can activate itself, but it has to be activated by something else first. What is it?
Dendritic cell?
Dendritic cell or a macrophage, right, can help activate the T helper cell. And so in this case, right, the B cell to become fully active needs help from a T helper cell. A T helper cell to become fully active needs help from a phagocyte, right? A lot of times it's a dendritic cell, but it can be a macrophage. and then it goes through a similar process. After antigen recognition and after activation, it can then divide and produce effector cells and memory cells, just like we see in the B cell. Okay? So again, this is showing you what's happening in B cells, but a very similar process is also occurring in T cells with a couple small differences, right? One of the most important ones being the activating cell. So we've talked about it a lot, but this is kind of that antigen recognition. One of the things to point out, and we'll continue to emphasize this, is I've mentioned before that the receptor that is presenting antigen to T cells is called an MHC receptor. That stands for major histocompatibility complex, right? And these receptors are going to be on the surface of cells that present antigens to T cells. All right. So here, right, it shows how, right, a helper or how a B cell can get help from a helper T cell. And so in this case, right, an antigen represented here by this Rubik's Cube binds to a B cell receptor. Upon binding of the antigen, that antigen is taken into the cell via endocytosis, right? So that's what this endosome is, right? It's then broken down like we would during phagocytosis, and it's presented on the surface of this B cell, right? And it's specifically presented in something called MHC class 2. MHC class 2 is going to be the receptor that is presenting antigens that have been taken up from the environment and then brought into the cell, right? And you'll see how that's different in MHC1 in just a second. Now, if the antigen that was recognized by this B cell is foreign, right? So here you see a microbial antigen presented. The TH cell, right, via its T cell receptor will recognize it and will deliver cytokines, right, an activation signal to that B cell, and then that leads to the proliferation of those plasma cells and those memory cells that we saw in our last chart. If the antigen being presented on the surface is harmless, right, then that means that B cell recognized something that it shouldn't. And so the T cell does not recognize it. It does not deliver that activation signal. And that B cell becomes anergic, right? Basically, it's just floating around until it dies. It's not going to do anything. Okay. So this is what's going on in that B cell activation step before we have proliferation of those plasma and memory cells. Okay. Does that make sense? Does anyone have any questions on this so far? No? Okay. So we'll kind of still revisit this idea. This is what I've mentioned before, and you can see here that the initial response, so the primary response, so this is the first time that you've seen a pathogen, right? And so the first thing you start to do is you start to produce IgM. So IgM is your initial response, and as you're producing IgM, you start to ramp up IgG, right? So IgG is going to be, in this case, the primary antibody that's going to remove this pathogen, right? And eventually here you see a kind of a small peak, right? And this small peak is going to be enough to remove that pathogen. But this is why in some cases it could take you days to a week, maybe even longer to recover from an illness, because your body, right, is building up enough antibodies in order to effectively remove that pathogen. But during this whole time, right, we mentioned this with T cells and B cells, you have produced memory cells. And so now the secondary response, right, or the next exposure to that same pathogen, because you have those memory cells present, you're able to produce a very large quantity of antibodies. In fact, you can produce so much antibodies that you can neutralize or often neutralize that pathogen before you even have symptoms, or maybe your symptoms will be incredibly mild because your response is so quick and so robust to that pathogen. All right, does that make sense? So that's kind of, again, I'm going to keep on emphasizing this point. This is the general principle of a vaccine, right? When you go to the doctor and you get that vaccine, what we're doing is we're inducing a primary response, all right? We are inducing the immune system to make memory cells so that when you actually see, or if you actually see that pathogen, the response that you generate, right, is so fierce that that pathogen does not cause serious illness in you. All right. And so, please note, if we're talking about pathogen, even if you had a vaccine, it's still multiplying in your body, right? It's still present, but your body is able to respond so quickly with such a large antibody load that a lot of times you don't even know that you've seen that pathogen again, or seen that pathogen naturally for the first time. So again, what you see here is a primary response. In this case, we're focusing on a B cell, but a T cell being the same boat. What I showed you 2 slides ago is, all right, how is activation, in this case, of these B cells occurring? And then, right, here you're fighting the pathogen, right? So this is still part of that primary response. But these memory cells that you formed here, right, are going to hang around and be a major part of that secondary response that is quick and produces a large load of antibodies to eliminate the pathogen before you have any symptoms, or maybe you only have mild symptoms. All right. Does anybody have any questions on this concept? This is a really important concept to understand, because it shows that you are able to recognize the different steps involved in antigen recognition. This is also important groundwork for understanding, again, the basic principles of how vaccinations work.
I have a question.
I have an answer.
So the only differences between the B cells and the T cells are the activation step?
Well, so another difference you could point out here, right, is a T cell is not recognizing free-floating antigen, right? Something needs to be presented to it, right? Because it can't recognize it on its own. So it has to have an antigen that's shown to it. And then once the antigen's shown to it, then you have a really kind of a similar idea here. So once the antigen's shown to that T cell, if it's foreign, then the dendritic cellular macrophage that's showing that antigen to the T cell will deliver signals that activate that T cell. And then that T cell will proliferate, form effector cells to fight the infection, form memory cells for later for that secondary immune response. Does that kind of make sense? Yeah? Okay, I'll find out if it doesn't. All right, deal? And some of this you may need to look through a little bit more. Is there a new message? I guess not. I thought I saw somebody typing them. All right, cool. Does that make sense though, Lauren? And so it might be good to, kind of make notes of some of those, differences, but the same general principle kind of applies. You have the immature, the naive, the activated, the effector cells, and the memory cells that are being produced during clonal expansion. Okay. So let's talk about T cells, and maybe this will provide some clarification. If it doesn't, we can go over it again. Right, so I already mentioned CD4 T helper cells are recognizing MHC2, and MHC2 is presenting external or exogenous antigens, right? So that means antigens that it's taken up from the environment, right? So presumably something has been phagocytized or consumed from the outside and is now being presented on these MHC2 molecules. Okay. Again, we're not going to go over these, but if you've ever read about T cells, which I don't know why you would have, but maybe you have, there are different types of helper cells. There are Th1 and Th2. Those are kind of the most widely studied T cells, but Th1 cells are important for macrophage activation, while Th2 cells are important for the antibody response, right, focusing primarily on B cells. T regulatory cells fall into the T helper cell category. You also have Th17, Th9, and there's a slew of other cells that have been identified out there, okay? But we focus primarily on Th1 and Th2, and I'm fine if you just refer to them as CD helper cells or T helper cells for this exam. You also have, right, your CD8 cytotoxic T cells, right? So that's what CD8 is. They are recognizing antigen that's presented on MHC1, right? And this antigen is going to be internal or endogenous antigens. So primarily the antigen that's going to be presented here, right, is going to be the antigen from viral infections, right? That's what they're primarily going to focus on, all right? So cytotoxic T cells target this antigen, and then they release granulocytes. They are granulocytes. I'm sorry. They release granules, which then kill the virally infected cell. One thing I want to point out, I'll try to keep on emphasizing this. Because MHC2 is presenting external or exogenous antigens, that means that you have to be capable of ingesting antigens, right? And so MHC2 is going to be found on dendritic cells, macrophages, and if you go back to a couple figures, even B cells, right? So B cell is not what we'd consider a classic phagocyte, but it is capable of taking an external antigen and presenting it, right? So MHC2 is only present on cells that are purposefully phagocytizing pathogens or antigens in order to present them. MHC1 is present, and again, there are some interesting exceptions in the body, but in this case, we're just going to say is present on every cell type, or at least we'll say present on almost every cell type, right? The way I kind of look at MHC1 is if you imagine each cell had a little window on it that you could look into that cell and see what's going on to make sure the cell is normal and healthy and doing okay. That's what MHC1 is. MHC1 gives our immune system the ability to survey every cell that's present in our body, okay? So here's just showing or highlighting that difference that MHC1 interacts with the T cell receptor on a CD8 or cytotoxic T cell. You see that here on the top. And so note my little colored blocks are inside the cell. Presumably, maybe this is a virus, right? That virus gets broken down by the cell, presented via MHC1, right, which can interact with the T cell receptor in CD8. And again, if this is formed, then this cytotoxic T cell will deliver a kill package to this cell, induce apoptosis, and that virus, presumably, right, will no longer be able to hijack or use the host cell machinery, okay? This is what we saw for the B cell, but again, let's go over it. So this could be, right, a B cell, a macrophage, or a dendritic cell. It internalizes in this case, the Rubik's Cube that's been floating around in your body. It breaks down that Rubik's Cube, presents different portions of antigens from it on its MHC class II receptors, which then can interact with the T cell receptor of a CD4 T helper cell. And if recognition occurs, right, then we'd have those signals that are delivered that lead to activation of whatever this cell type may be, either T cell, B cell, or dendritic cell. All right.
So I have a question.
7.
So, the CD8 T cell receptor are on cytotoxic cells, and the CD4 is going to be on the T helper cells.
Yes, ma'am.
Okay, and then I was also, so it looks like MHC1 is on like the surface of the cell. Like I thought it was supposed to be like inside. Like, I'm so confused about that.
MHC1 is the receptor. The MHC, no matter if you're 1 or 2, MHC, major histopathology complex, is going to be a receptor that's on the surface of the cell membrane that's presenting antigen to a helper T cell. Because remember, helper T cells can't see free-floating antigen.
Okay.
When you have receptors that are not on the surface, that would be, I don't know if maybe you're referring to like a T cell receptor or a B cell receptor in immature cells that aren't expressing that receptor on the surface yet. Is that maybe what you're talking about?
Yeah. Okay.
And Noah, I mean, please feel free as I go through this to interrupt me again. If something starts to click, right, or you're like, you need to revisit something. But I think that's probably what you're referring to if you're referring to something not being on the surface. I mean, if we really wanted to get, let's see, I'm trying to combine the word cytology and philosophy. Psi-philosophology, I don't know, right? Really get deep on the cell ideas is that, you know, every receptor is produced inside of the cell and then is eventually targeted for the cell surface somehow, right? So if you wanted to go really deep on me, you could be like, you know, everything that was on the surface was ones inside the cell. But I think that's what you were referring to that we were talking about earlier, is that in an immature cell, whether it's a T cell or B cell, it's not expressing this receptor on the surface yet because the receptors are being made and haven't gotten to the surface, right? So if we need clarification, right, just interrupt me again as we move through this, all right? So for activation of these cells, I think what you can probably appreciate or hopefully appreciate is that as cells are moving through circulation, that's not a good way for them to be activated. Again, the sheer force that's carrying these cells through your circulatory system does not really allow prolonged interactions. So, right, this is where lymphoid organs come into play, right? And lymphoid organs are bringing T cells, B cells, macrophages, dendritic cells, right? All of these things together in one place where they can test things out and where they can be activated. Once they've tested things out and they're activated, they can leave the lymphoid organs, enter circulation, and then go to the target area or the target tissue where they're needed, all right? So some of your primary lymphoid organs are going to be the bone marrow and the thymus, right? The bone marrow is where all, right, cells of the immune system are produced, whether it be innate or adaptive, right? When we talk about the adaptive immune system, right, B cells are produced and mature in the bone marrow, while T cells, right, reach their final maturation point in the thymus, okay? And so if you remember, or if you've had ANP2, or maybe you did some pig dissections, I don't know, in high school, right? The thymus essentially kind of sits almost right on top of the heart, right? So the thyroid's up here, and the thymus is kind of down here, almost right on top of the heart. This is the place where T cells go to learn. And what's interesting is that once you kind of hit your teens to your early 20s, a lot of that thymic tissue gets replaced with fat. And the idea is that you already have enough of a T cell repertoire to fight a lot of the infections that you'll see in the present and in the future. But it's also one of the reasons is that as you age, right, the immune system of your 80 to 90-year-old grandparent or great-grandparent is not as effective or efficient because their thymus is no longer active. All right, so kind of an interesting little evolutionary tidbit there. For your secondary lymphoid organs, right, tonsils, adenoids, spleens, or not spleens, hopefully you only have one spleen, right? Your lymph nodes, and so if you've ever been, right, to the doctor, oh, like Noel's family, this would be like a great one probably with all the strep throat, you know, that they get. You know, a doctor, when they go to visit them, or a PA or a nurse practitioner, whoever you're going to see, right, one of the first things they do if you're complaining about that is they'll kind of come up here and they'll feel around, right? And if your lymph nodes are swollen, that indicates that your immune system is actively trying to fight something. And so it doesn't allow for a diagnosis of exactly what's wrong, but it does give, you know, a doctor a pretty good indication that, hey, something's actually wrong with Noelle, or presumably it sounds like often one of her brothers, right? How many brothers do you have?
I have 4.
You have 4. There we go. Okay, good. One of her brothers, right? And so it's just kind of a quick little thing. Any of you are interested, like in the plague, right? The bacteria that causes the plague loves to hang out in lymph nodes. And so you have axillary lymph nodes. So those lymph nodes will get really swollen. You also have lymph nodes in your groin. Those are kind of some of the major hotspots of lymph nodes. And so those lymph nodes will swell and will sometimes even burst in people that have the plague. So, I'm sure you didn't want it before, but hopefully that's even more motivation to not get it. All right. And so these lymph organs and these primary and secondary lymph organs are really important because, again, this is the spot where all of these cells are brought and come together. All right. And again, it allows an environment that is safe for testing and safe for expansion. And then after testing and expansion, they can leave the lymphoid, or they can leave the lymph system, enter the circulatory system, and go to their target tissues. Right? We've already talked about this, but again, it continues to be an important concept to realize that CD4 T cells are recognizing MHC2, CD8 T cells are recognizing MHC1. This is another important diagram because it shows you, right? I keep on mentioning, right, the importance of macrophages and dendritic cells. And I say, hey, dendritic cells do this, macrophages can do this too. But no one can do it quite like a dendritic cell, right? So that's why you've seen on my notes, and you may see in your textbook, right? When we talk about a professional antigen-presenting cell, all right, no one does it like a dendritic cell. Because what dendritic cells like to do is they like to activate CD4 and CD8 cells at the same time. All right. And so this is speeding up the immune response, right? The fact that you can activate both. And so, right, if a cell needs to be destroyed or killed because there's a virus present, well, you want the CD8 cells activated for that. That's awesome. But you also want CD4 T cells activated. Why would you want CD4 T cells activated if you're dealing with a viral infection?
Because like a viral goes in and overtakes the cell to make its own protein.
It's true. No. And that's exactly what a virus does, Rachel D. So I like that. But a CD8 T cell, right, is going to go and kill that, right? It's going to go kill that virally infected cell. So what do we need CD4 cells then if we're talking about a viral infection? I'm causing you to try to kind of pull a couple things together. So don't worry if we're not quite there. Who was saying something?
Do they use cytokines to activate more killer T cells and B cells?
Okay, perfect, right? So they're going to produce cytokines that can activate more cytotoxic T cells. So that's really important. And then you said cytokines that activate B cells. Why do I want a B cell activated during a viral infection? You're right. I 100% agree. But why? What is a B cell doing that would help me with a virus?
Creating antibodies.
Okay, good. And then what do the antibodies do to the virus?
Inactivate it, keeping it from infecting other cells.
Right. So they neutralize it, right? That's one of the things that antibodies do. Antibodies neutralize the virus. You also, right, if you go back and you look at antibody-dependent cellular cytotoxicity, ADCC, right? That fancy kind of big word there. You can see that those antibodies can coat the outside of a virally infected cell and allow natural killer cells to also join in on the fun. All right. And so here you see this is really kind of impressive because this dendritic cell is helping bring the whole immune system into the kind of the game, right? Getting everybody involved to help try to clear that infection, right? So dendritic cells are really good. At this, here you see, right, this is activation. Here you see, right, a dendritic cell that is actually expressing self-antigen. Remember, dendritic cells are just sampling. They're grabbing onto everything. Some of it's foreign, some of it's part of the host, and then they go and present it in a lymph organ, and they have the T cells and the B cells decide, right? And in this case, if a B cell or T cell responds to something that a dendritic cell is taken in that's not foreign, well then again, these cells become anergic and they're not responding because, or they're become energetic, so they can't respond because we don't want them to start to kill self. All right. So we gotta love our dendritic cells. All right. So let's continue. I don't know if maybe you're picking up on a theme or something that could be a good test question, because I seem to really love talking about antigen presentation and activation. And so here, right, again, you see some type of normal cytoplasmic protein, right? And again, we talked about this idea, we visited this idea of remodeling, right? And so even the proteins that are good inside of your cell are constantly being torn down and built back up. Well, when they're torn down, they get presented on the surface. And a good CD8 T cell will not recognize normal cytoplasmic proteins that are presented on the surface of the cell. All right? So this is a T cell that has been educated really, really well. Now, if this is foreign, we'd want that T cell to recognize it. But if it's not, we want the T cell to leave it alone. So that T cell gets to stay around in search of its antigen that it's supposed to recognize that is presumably pathogenic. Here, right, you see, this case, this is what it looks like with a pathogenic antigen, which we've seen this before, but this is some type of viral protein is presented on the surface. The CD8 T cell recognizes it via its T cell receptor, says that's bad, delivers, kill package. You can also call it death package, whatever you think sounds more awesome or deadly, all right? So it delivers this death package, induces apoptosis, as you guys have mentioned to me before. If the cell's dead, it can't make new viral particles, okay? Here, I'm not gonna spend time on this, but again, this is T helper cell activation. Remember, in this case, we use the example of a B cell, but this could be a dendritic cell, or this could be a macrophage that is also presenting an antigen on its cell surface. One of the kind of important things that I want to point out is that, and again, there's always exceptions, but when we're talking about MHC1, MHC1 is typically always going to have an antigen present in it because that cell is always undergoing remodeling, and so there's always going to be probably a host antigen present in the MHC1 receptor, or, right, if it's infected, then it would have that pathogenic antigen present in its receptor. MHC2, right, for the most part, right, a B cell should not recognize self, right? It can, but, okay, let me take this back. So a B cell, a macrophage, and dendritic cell, they have to eat something or purposefully take it in to present it, right? So for the most part, antigens that are being presented on the surface, right, of MHC2 are typically going to be pathogenic, all right? Again, it's possible that a B cell could take in a self-antigen, it could present it, and hopefully it's not recognized. But the majority of antigens that are presented on MHC2 are going to be pathogenic, right? Whereas opposed to antigens that are presented on MHC1, right, could be one or could be the other because you're always presenting some type of antigen because you're always undergoing remodeling, right? So here, right, that was a B cell. Here you see macrophage, but a very similar process. Taking the Rubik cube via phagocytosis, you then break it down, right, and you present it on your surface. If it's recognized as being foreign, right, you deliver some type of activation signal, right, via cytokines, right? These cytokines activate this macrophage, right? If there's any other cells in the area, they also encounter those cytokines and they become activated, okay? You know, just a quick little side note, this is also the reason that we have some autoimmune diseases or disorders is because, you know, all of these cells that have different receptors and the ability to recognize different things are hanging out in the lymphoid organs, right? So they're all in close proximity. And so let's say this macrophage presents molecules that are foreign, right? This is a pathogen. It's recognized here. It gets activated. But there could be a T cell in the area that actually interacts with host molecules that would normally die, but it happens to be in the general vicinity, and it gets activated by some of these cytokines. And so it's almost like collateral activation or peripheral activation. The signal actually wasn't meant for it, but it interacts or encounters that signal, becomes activated, and then can interact with host antigens that it shouldn't be interacting with. All right. So natural killer cells are pretty cool because one of the things they do is they kill cells that lack MHC1. One of the things we didn't really go over, and we don't have a lot of time to go over because there's just so much, is that some viruses, when they take over the host cell machinery, they actually prevent MHC1 from being expressed on the surface of the host cell. And so the basic concept there is if MHC1 is like a window that lets you look into the cell to see what's going on, the virus says, well, I'm going to block or close that window. And if I block or close that window, CD8 T cells can't interact with that cell. And so the virus in that case has kind of eliminated the threat of a CD8 T cell inducing apoptosis. And that's where natural killer cells kind of come in and pick up the slack a little bit. They come in and they're like, okay, that's fine. MHC1's not present. MHC1 should be present on pretty much every cell in the body. So if we don't see it, we are going to, right, induce apoptosis, right? They also have, right, on their surface, right, receptors for FHC, right, that constant region of antibodies. So that's where ADCC comes into place. And again, upon activation, whether not seeing MHC1 or seeing a host cell covered with antibodies, they release cytokines that induce apoptosis in that cell. All right. And so this is just showing that process, right? So the virus infects that cell. Here you have MHC1. At this point, this is host antigens being presented, right? So a T cell wouldn't do anything to that. The virus comes in, and when the virus comes in and starts multiplying, it inhibits the expression of MHC1. So a CD8 cell can't interact with it. However, a natural killer cell notices a lack of MHC1 expression, delivers a death or kill package, which then induces apoptosis, and that cell can no longer be used to replicate viral particles. Okay? So this is an overview of the entire immune system. All right. Of course, it can get more complicated than this, but I do like this because it shows clonal expansion of T cells and B cells. It shows the interaction between the innate and the adaptive immune system and how that leads to the neutralization and removal of pathogens or infected cells. All right. So this is kind of a good summary chart. Again, you know, if you just glance at it, it seems kind of overwhelming, but if you start to kind of follow the arrows, it really does start to make sense and kind of pull everything together. All right. So let me see what I have here, if there's anything here. I'm gonna skip this section, right? This is talking about pathogens and infectious disease. I really feel like we're gonna go over this a lot next week and even a little bit tomorrow. So kind of ignore this. What we will do really quickly is talk about how pathogens infect, even though we've gone over this, you know, before. And so in this case, right, they have to be able to adhere. We've talked about this before. Fimbriae on bacteria bind to certain receptors. Spikes on viruses bind to certain receptors and allow attachment to specific cells, right? And so in this case, these receptors that they're binding to are often important for physiological functions, or are important for physiological functions of the cells that express the receptors. And so bacteria and viruses are able to take advantage of the presence of these receptors. And again, this is why we see tissue specificity a lot of times in what pathogens are infecting or where we're finding pathogens in the system. Remember, we briefly mentioned with SARS-CoV-2, right, the COVID-19 virus, that it expresses receptors expresses spikes for a receptor called ACE2, which is found in respiratory tissue and in gastrointestinal tissue. And what do you know? Some of the virus symptoms are associated with the respiratory system and the GI system, right? So we have to adhere. That's how we have to establish affection. right? We have to be able to colonize and multiply. So in this case, right, this is where biofilms become very important for bacteria as they multiply. In the absence of biofilms, right, the body can be a pretty harsh environment. There are constant threats that are trying to eliminate the bacteria. So biofilm is a good way to try to protect bacteria from the immune system. It requires a lot more effort and time to remove biofilms. Right, this is not showing a virus, but again, if we're talking about establishing an infection in a virus, that means the virus has to enter the cell, take over the cellular machinery, and make a bunch of copies of itself. All right, so although it looks different, a virus is still doing the same thing, except the virus is doing this on an individual cell level, right, utilizing your cells. And maybe I should have put this first, because before any of these things can happen, the barrier, right, that first line of defense has to be breached. And those two main lines of defense are going to be your skin and your mucous membrane. The skin is the hardest barrier to breach. The only way pathogens are getting in if there's some type of damage to your skin. For the mucous membrane, this is actually the most common entry point for viruses, right? So presumably, if somebody with the COVID-19 virus came and coughed on your arm or drooled on your arm for some reason, right? As long as you have no cuts, right, that virus is not going to enter through your skin, right? The COVID-19 virus needs to enter through mucosal membranes, or I haven't really looked at entry point via gastrointestinal membranes, but presumably that's also possible. What's really cool is scattered throughout your digestive system, you have these things called M cells, and M cells are basically taking in small bits of antigens to sample for the immune system to see if there's some type of infection. And some pathogens are actually great at tricking these M cells to letting a whole pathogen cross, all right? And so it's a really kind of cool adaptive mechanism. Some pathogens actually purposefully let dendritic cells grab onto them so that they can carry them to the lymph nodes where they can infect lymphocytes, right? So there's a lot of cool mechanisms that pathogens have developed to get around mucous membranes. This is primarily going to be for bacterial infections, but you do have some bacteria that have something on it called an ejectosome. And an ejectosome basically delivers, right, effector molecules, and they can do a variety of things, but they make the host cell, it almost looks kind of like a syringe here, right? They make a host cell more susceptible to infection, all right? And so you're basically priming that host cell, you know, for an infection. But even establishing the infection, remember I talked about with the COVID-19 virus, those spikes actually have to interact with transmembrane proteins on the host cell before they can become fully activated to allow attachment to their target cells. And so there's a lot of different things that pathogens can do to get primed for an infection or to prime their target cell for an infection. All right. And so the last thing we're going to talk about here, and then we'll take a short break, is some pathogens have gotten really good at evading the host, specifically evading phagocytosis, right? And so how do you do this? You do this in three basic mechanisms. You prevent encounters with the phagocyte, right? If the phagocyte doesn't see you or interact with you, it's not going to eat you. If it can't recognize you, even if it's close to you, or it can't attach to you, it can't eat you. And in some cases, right, and this is where I think it's Allie and I can't remember who else are going to talk about TB. Some bacteria are great, or some pathogens are great at surviving within phagocytes, and Professor Perry even mentioned that certain fungi can do this. And you can even say certain viruses can do this, right? Certain viruses can incorporate themselves into the genome of a phagocyte, right? So you have to prevent encounter, avoid recognition and attachment, or survive within phagocytes. If you do one of these three, then you avoid getting eaten. And so here, this is just kind of a visualization of this. How do these different things happen? Prevent encounters. Well, remember, C5a is a chemoattractant. So some pathogens can secrete C5a peptidase, right, which breaks down C5a. And so basically you wipe away the bread trail or the breadcrumb trail that would lead a lymphocyte to you, right? You can also release membrane damaging toxins. So think about this again. We'll do Glory for this one because I haven't picked on Glory. Let's say Glory is involved in a high-speed chase from the cops. There she is, right? And so one of the things she may do is, if you've ever ridden with Glory, I assume that you will notice that she keeps like a box of tacks in her glove compartment, right? So she tosses those out the window. If she knows a cop's chasing her, it pops the cop's tires. If the cop's car can't drive because it doesn't have tires anymore, well, then Glory gets away. And the same idea here. Membrane damaging toxins are essentially popping the tires, or in this case, maybe even killing the phagocyte that's coming after them. All right. Avoiding recognition, right? Remember, we talked about capsules. It's kind of a sticky shell that's around the pathogen that prevents interaction with its cell wall or cell surface. M protein is, I think, neutralizing. I have to go back. I'll see. I have this next slide. I believe it neutralizes some of the complement proteins, prevents activation of the complement system. Some pathogens actually express FC receptors on the surface. I'll show you what that looks like in just a second. And then survive or escape within a phagocyte. So here, right, some pathogens, once they're ingested, can actually escape from the phagosome. Some pathogens can prevent fusion of the phagosome with lysosomes. Remember, lysosomes contain all of those enzymes and those acids that kill pathogens. And so if that lysosome can't fuse with the phagosome, then the pathogen doesn't die. And then even after fusion, right, and this is where tuberculosis and at least one of the types of fungi that Professor Perry talked about, they can actually survive these harsh conditions, all right? And so in essence, it's impossible for the cell to digest them. And that's when you see the formation of those giant cells and those granulomas to try to wall that pathogen off from the immune system, all right? So this is everything that we just went through except the text, right? Yes, M protein binds to the complement proteins. Got it. I'm glad I know that since I'm teaching it. And so just a couple different ways that you can survive there. When I said FC receptors, how's that advantageous? Well, here, right, this is what a pathogen that's encountering antibodies normally looks like. The antibodies bind to it, and then those different things that we talked about antibodies doing, which are usually bad for the pathogen, can happen. However, if the pathogen has its own set of FC receptors, it then binds antibodies and they face out and they actually start to resemble some type of host cell, and it allows them to evade the immune system. Phagocytes don't bind to the variable regions, and so they will have a lot more trouble interacting with a pathogen that essentially has turned around the antibody to face outwards. Okay. Evading the host, surviving with the phagocytes is what we just talked about. We already talked about all those details there. Here are a couple other alternatives, right? Some pathogens produce proteases that break down antibodies. We talked about antigenic variation, so altering the expression of surface antigens to avoid antibodies and recognition by the immune system. And then often, right, we'll see pathogens mimicking the host by being covered with host molecules, right? Viruses can do this, right? An important part of that envelope is usually part of the host membrane, and so it's harder to recognize. Some pathogens will actually express host-like proteins to put on their surface so that they can look more host-like or disguise themselves. We're not going to really talk about, this is just some of my research. You can look at that. We've been talking about the microbiome throughout, so you can ignore this last part unless you're really interested in some of the stuff that I used to do research on, which I think is fascinating, but we're not going to take class time for that at the moment. Maybe we have extra time, we can go over some of that. So it's 10:15. Let's take a short break just so I can get up and walk around. So 10:15, let's come back around 10:20 so we can jump right in and I can get you guys out of here. All right, so group 3 will be presenting, so be ready for that. Just a short break was 10:16, 10:21-ish, right? So try to take a solid 5 minutes and then we'll hit the ground. Megan, I'm excited to start some research projects with you where we use fungi to grow different things or to make different things. Yeah, your game?
Yeah, I don't know. It seems pretty cool.
Okay. We'll talk about it in the fall. All right.